Hello, in this video we will go through an introduction to Biopython. Okay, so these are the content. Okay, we'll go through the Biopython. What is this Biopython project about? How to pass a sequence file formats is so-called like load now. Okay, sequence object and biological methods on sequence object. Okay, this Biopython project, okay, is International Association of Developers, okay, of freely available Python tools for computational molecular biology. Okay, so we're gonna use this BioPython, this package, to help us do a lot of analysis. Okay, computational molecular biology. Okay, so the resources are, you can go to website, okay, biopython.org. Okay, or for this term we will be following this website. Okay, and inside there's a cookbook. Okay, tutorial is so-called a cookbook, so-called like a textbook. Okay, that we follow. Okay, so what does it include parser. Okay, parser is like a file loader. Okay, with various bioinformatics file formats. Okay, because when they record the bio data, okay, the sequences, okay, usually they will record in terms of FASTA or GenBank files. There's other files as well, but in this subject, okay, this module, we're just going to cover mostly just FASTA and GenBank file. Okay, access online services, okay, NCBI and Anthrax, okay, and PubMed database. Okay, we will be using interface to common biological programs like BLAST. Okay, so how to check your BioPython package? Okay, just follow the installation video that we send you on term 1. Okay, and make sure you install them. Okay, so these are the two steps that check that what version you are using now. Okay, the version, as long as it's around 1.6, then should be fine. Okay, but if you encounter error when you type this import bio, okay, then you please watch the e-video on how to install the BioPython and just install it. Okay, so first of all, we're gonna pass some sequence file formats. Okay, what pass is just like loading. Okay, as I said, loading file formats. Okay, we will use this bio.seqio. Okay, means sequence input output. Okay, yeah, to pass file format. Okay, we want to pass this file format into a format such that we can manipulate them with the Python language, like find the reverse complement and stuff. Okay, so we will go through two simple examples. More will be covered in, I think, the next few lesson. Okay, that how do we convert from FASTA to GenBank or more advanced parsing. Okay, so a simple one. Okay, for example, download this file. Okay, this FASTA file. Okay, you can find this on your Olive. Okay, for this example. Okay, this FASTA file contains 94 records, so 94 different sequences. Okay, which start with a line of the greater sign, okay, which followed by the ID and the description. Remember what we went through in term 1. Okay, FASTA file, the format is like that. They will have a greater sign first. After that, okay, greater sign first. After that, the name and the description, ID number and the description. Okay, then followed by the sequences, the sequence. Okay, so there's 94 of them. Okay, in this file. Okay, so how do we pass this file? So how do we load this file? Okay, import from bio, import this sequence IO. Okay, seqio. Okay, then this is a for loop lah. Okay, for this one is up to you. Okay, you can use I. Okay, for sequence in seq underscore record in this pass of. So what they do is they use this module and pass the file of this ls underscore ocit.fasta. Okay, then you must write down over here. Okay, this is a file name. Okay, then this is the format that you are using. Okay, you are passing. Okay, for this one is FASTA lah. Okay, and over here just key in your location of your file. So different, if you got a different file location, just key in accordingly. Okay, then what this do? It print the ID, print the sequence, print the length. So this is what you will have. For example, the first one, you look at this and look at this. Okay, So they will print out the ID, okay, the description. We want the ID, okay, the just not the code, just on ID, the sequence, okay. Okay, the ID will give you the ID lah. Okay, dot sequence will give you a sequence. Then this is the length of the sequence, 740. So you see this, the sequence. What is the sequence? Okay, so these are the sequence. So how long? What's the length? Okay, 740. So you do all for the 94 of them. Okay. Then same for the GenBank. Okay. Download this file and then the same thing, the 94 record. Okay, just that the GenBank compared to the FASTA file, GenBank file will have more information as compared to the FASTA file. As you can see from over here, you can download and open using your notepad, and you can see that the information they give is much more. And we will look at all this, okay, in the next lecture. Okay. So same thing to pass it. Okay, everything is the same, just that this one changed to GenBank. Okay, and then the same thing, location of your file. Okay, please don't type location of file. Please type based on whatever you save, where it's saved at. Okay, then okay, ls underscore orchid.gbk. Okay, this is a file format which is GenBank. Okay, so over here. Okay, so what's the difference? It's just the ID. Okay, if you look back, okay, the ID for in terms of FASTA is like this, so it's long one. But what's important is just this number. Okay, so GenBank will straight away give you the number straight away. Okay. Okay, yeah. So the rest is the same. Okay, if you notice, there's also another difference, which is over here. You see this single letter alphabet. Okay, and this one is IUPAC ambiguous DNA. Okay, what is this? This is called alphabet, which will introduce this in the next few slides. Okay. So now, for Biopython, it uses sequence SEQ object to deal with sequences. They are basically string, okay, like Python string, just that they format it nicer. Okay, what are the two important differences you can find between the sequence object and Python string? Okay, for sequence object, okay, between these two and standard Python string, okay, biological method which we're going to introduce later, okay, like translate, reverse complement may produce different output based on whatever sequence you have. Okay, and sequence got an important attribute alphabet, okay, which is an interpretation of how the individual character made out of the string. Okay, what is it? What am I talking about? We'll cover it in the next few slides. Okay, so we introduce this alphabet now. Okay, as mentioned, sequence object has a important attribute alphabet. Okay, which currently all current available attribute alphabet will be inside this module. Okay, called bio.alphabet. Okay, they use IUPAC alphabets. Okay, so it's using this. Okay, then provide that basic definition for protein, DNA, and RNA. So basically, it's just to give a category to your sequence. Is your sequence a DNA? Is it an RNA? Is it a protein sequence? So this is just a category for it. Okay, so that when you give a category, next time when you do the reverse complement and stuff, it will be faster lah. Okay, will not have error. Okay, they help you do accordingly. Okay, so these are a few I summarized for you. So for protein representation, IUPAC protein is the basic protein class, or the letters. Extended IUPAC protein, okay, include U, O, B, Z, J, and X accordingly over here, described over here. So it's extra, they'll give you additional letters as compared to the basic letters. Okay, DNA representation, there is 3. Okay, IUPAC, unambiguous DNA, and ambiguous DNA. Unambiguous DNA is just, they would only take in these 4 basic nucleotides. So nucleotide base, okay, so ATGC. Ambiguous, okay, allow ambiguity letters for all possible situations. So any of outside of these 4 letters, they can. Okay, extended is allowed modified bases. Okay, then RNA representative also the same, unambiguous, ambiguous. So unambiguous is AUGC. Okay, then ambiguous is any ambiguity letter can be used. Okay. Why is this important? Later when we use the biological method, you'll see, okay, why is it that important? Okay, so first let's try to create a sequence with a generic alphabet. Okay, all you need to do is from bio sequence import sequence, then you put my sequence equals seq over bracket, then type in a sequence and close it. Okay, then yeah, so my sequence, if you key in like that, it's just in. If you never specify the alphabet, it will give you a generic alphabet, which is just alphabet itself. Okay, so note that you can change normal string to sequence object, and you can change the sequence object back to normal standard Python string. Okay, by just use a str, then your my sequence. Okay, this one is what we learned in term 1. Okay, suppose you want to create an unambiguous DNA alphabet. Okay, so you other than import the sequence, you must from the alphabet bio.alphabet import the IUPAC. Okay, from there you put, instead of just equal to sequence, then write down the sequence, then you close. Okay, you add in this line. Okay, this argument as well. IUPAC dot unambiguous underscore DNA. Then over here, when you key in again, then you show this is unambiguous DNA. Okay, so this one means that your sequence will be a DNA sequence, and it will take in the ATGC. Okay. So sequence object is just like string. So you can do sequence object like the normal standard Python string lah. Okay, so you can so-called slice them. Okay, you want the whichever element. So this one, the second 0, 1, 2, the T will come out. Okay, you want to count number G, you can length. It's all the same. You can slice them as well. Okay, and furthermore, you can use bio.seq utils module to help you find the GC content. Okay, by the GC function. Okay, all this you can go and Google and find out what are the modules that can help you find things faster. Remember last time when we do the when without using BioPython, okay, we need to ourselves create the GC content function itself. Okay, yeah. So over here, they already help you create everything already. So all you need to do is just import the correct module and the correct function, then you run accordingly. Okay. Okay, and what if you want to concatenate two generic alphabet sequences? Can. Okay, so for example, this is a protein sequence, this is DNA sequence, so you want to add these two up. Okay, protein sequence, these are letters, and they add these are the thing lah. However, and note that okay, Python string you can use the dot join method, but sequence don't have, currently don't have. Okay, maybe if at the later version you may have. Okay, then okay, look at this example. However, you know that by right protein sequence and DNA sequence, you cannot join them together because they are two different things. Okay, so usually what we do is, okay, specify the appropriate alphabet. Okay, then an error will occur if you add sequences of incompatible alphabet. For example, okay, so import this and import the sequence. Okay, you know that this is a protein sequence, so you specify the alphabet as protein, IUPAC.protein, and this one you know there is a DNA sequence, so you underscore this. So you try to add them out, you have error accordingly because they are incompatible. Okay, so this is just to like for you to double check. Sometimes you doing some code, then you want to double check whether you want to add something up, then here will give you error. Okay, that's why it's very important for you to specify what category your sequence is. Okay, there are cases where two sequences are different alphabets but they are compatible. For example, okay, this is the alphabet module got other than a IUPAC types of naming of category. There are also other like generic nucleotide. So for example, this one generic nucleotide, this is your sequence, you use generic nucleotide. Okay, then this DNA your alphabet is use this. Okay, add these two up, they will give you a nucleotide alphabet lah. Okay, the generic one. Okay. Okay, biological methods on sequence object. Okay, why BioPython is so useful? Because instead of writing all the function from scratch again to find the complement, reverse complement, transcribe and translation or whatever, the BioPython package straight away have all these function. All you need to do is call as a not function method. All you need to do is just call the method, then everything will be okay. So complement, okay, we learned before, return the complement of the sequence. So change the A to T, change the C to G, and vice versa. Reverse complement, okay, to find the reverse complement of it. Transcribe is to change all the T to U, okay, and change the alphabet to RNA. Later we see example. Translate is translate mRNA sequence into a corresponding protein sequence. Okay, we're going to look at example. Okay, for example, we want to find the complement or reverse complement of this unambiguous DNA sequence like this. Okay, all you need to do is just dot complement and everything. Okay, G will change to C, A will change to T and stuff. Reverse complement, they will reverse it and find the complement. Okay, this if you want to find the reverse complement and complement of the DNA sequence. Okay, however, if you want to find the reverse complement or complement of protein sequence, error occur. Okay, why? Because protein sequence don't have reverse complement or complement. So over here we have an error. So here is this function is very good because they will help you check whether you can run the reverse complement or not. Okay, method I mean. Okay, transcription. Okay, remember last time what is transcription? Okay, so let's recall back the actual biological transcription process work. Okay, from template strand. Okay, so you know DNA sequence came in two pair base. Okay, so one is they always run from the 5 to the 3 end. Okay, so this is the coding strand, then this will be a template strand. Okay, run from here to here is the coding strand, then here to here is a template strand. Okay, DNA template strand. However, when they transcribe into a single stranded mRNA, what they do, okay, they don't use the coding strand, they use the template strand. Okay, then they so-called reverse complement it. Okay, then after that transcribe it. Okay, transcription, you get this. Okay, so but this is actual biological transcription. How does it work? Okay, however, for Biopython and most of the bioinformatics in general, we will work directly with the coding strand straight away and switch the T to U, which is much faster. Okay, over here, right, if you want to do by the code, if you do by the template strand, you need to find, over here they give you this, you need to find the reverse complement, then you need to transcribe to get to this. However, what it actually boils down to, actually you see this strand, the coding strand, you change all the T to U, then it's mRNA already. So in Biopython and in bioinformatics, okay, we work directly with the coding strand, is much faster. Okay, so yeah, okay, so this is the coding strand. Okay, so transcribe. Okay, okay, so we this coding strand deal is transcribe it, so this will be the mRNA. Look over here, the method is quite good. Why? Because initially you got a DNA sequence, correct? Okay, after that, when you transcribe, you know that after transcription is a mRNA sequence, right? Then they will change it for you. Okay, so they change all T to U. Okay, on the other hand, if only the sequence of template strand is given to you, then no choice. You need to find the reverse complement of it, then you transcribe it. Okay, so this method of transcription, they only help you from change the T to U. Okay, they will not help you do the reverse complement. So you need to do the reverse complement, then okay, you can do the transcription. Okay, so note that this Biopython also have this back underscore transcribe method, which will change your mRNA back to your coding strand. Okay, as I repeat again, this transcribe method will not okay, do the reverse complement for you. So they will straight away change the T to U. That's all. Okay, then back transcribe will give you from U to T straight away. So this is mRNA. Okay, then your alphabet is unambiguous RNA. So you back transcribe. Okay, it will give you the U change to T, and it becomes DNA. Okay. Okay, then lastly, translation. Okay, translation, you can translate an mRNA into a corresponding protein sequence. Okay, so they will look through. Okay, so same thing. Okay, 3 by 3, remember how we look, okay, in frame, so we from frame 0, 3 by 3. So AUG is the start codon. Okay, so it's represented by M. Okay, for the table, what letter to be used, you can use, you can go Google, or during tutorial I will show you. Okay, so AUG, so 3 by 3, they're going to just translate straight away to give you this, where star is the stop codon. Okay. Okay, and what's so good about this translation, they can directly translate from the coding strand. So you don't need to change it to U Okay, if you got the coding strand and you want the DNA sequence, you straight away you have this DNA sequence, just translate it. The result will be the same. You see this and this. Okay, so this is very good. So that's why the alphabet is very important. Okay, so if your alphabet is correct, okay, they will translate it accordingly. This RNA, they will use the translate, so they should translate to the correct one, and same over here. Okay. Okay, note that there are optional arguments that you can key in. For example, if you want to specify, you just want to translate up to the first in frame stop codon. Okay, what you can do is, this is the original one, correct? Okay, you put an optional two underscore stop equals to true. You'll stop at the very first codon, so you stop over here. Okay, and remember, when you translate, just now I forget to say, they will change to the alphabet to protein. Okay. Okay, there are different translation table in Biopython. Okay, by default translation will use standard genetic code. Okay, so however, if we do with mitochondria sequence, we need to use a different translation table. Okay, if you know the bio part lah. Okay, so for example, if the translation table, I'm not looking at the genetic one. Okay, standard genetic code, we use the mitochondria table. Okay, you need to specify which table you use by this optional argument. Table equals to 2 or this mitochondria table. Okay, so over here, some of the sequence I think will change accordingly. Okay, so either use either one. So if more information, you can look at on what is this different translation table. You can go to this website and go and look at it, or during tutorial I will show you, I print out for you. Okay, what does the table looks like, what letter represent what. Okay, so yeah. Okay, so this comes to the end of this lecture. Okay, so yeah, that's all. Thank you.